there's a few things that I thought about. I actually initially wanted to be a veterinarian. I liked animals and I liked biology, but I think after a while I realized that like being a vet wasn't exactly what I wanted to do, but I knew I liked biology. I kind of thought about teaching as well, so I think I went into college thinking that I was going to be maybe a teacher in biology, like for high school or maybe college. So I went to the University of Minnesota here for my undergraduate degree. So I got a undergraduate degree in biology, and then I went to University of Illinois at Chicago and got my master's degree there, and that was in molecular biology. And then after that I ended up working in a research lab at Northwestern University in Chicago for 8 years, and then moved back here and have been working in this lab for about 14 years. Well, I think biology in the beginning, was probably when I was younger, and I liked being outdoors and watching, ants, or watching, just watching things grow, looking at plants. How do they grow? Like, how do they change? And but then when I got into college and took classes, it was really fun seeing like the next level down, like the cells, like how do the cells interact, and what kind of signals happen between cells to tell like a plant to grow. How does a plant know to make a leaf and a stem, and likewise with animals, like how do the cells that start out as a single cell turn into an organism and create a leg or limbs or heart or liver? Like what messages are going on to like make that happen? So I've always been fascinated amazed with biology. Molecular biology is kind of the area, and cell biology, genetics, development, it kind of all works together. And so I study a particular protein that is involved in regulating gene expression. So if it goes wrong, there's a human syndrome, the developmental syndrome, that actually when our protein of interest is mutated, they have a lot of things go wrong during early development. So when they're in utero, like growing as an embryo and fetus, things go wrong, and they have small eyes, they have brain defects, they have digital, like their fingers fuse together or stay fused together, they have heart defects. Our goal is to kind of try and figure out how does our protein interact in those tissues, because each tissue is made up of different types of cells. Like eye tissue is different than heart tissue. So how is our protein playing a role in all these different tissues? Well, it must be able to kind of interact with different things. So we want to understand how it functions on a molecular level, because usually proteins, molecules interact with each other to do a job. And so we're just trying to figure out what our protein's role is in these different tissues. We've created a mouse that we can control the gene to knock out in certain tissues. So our gene of interest gets knocked out, say, in heart tissue as it's developing, and then we study whether taking away our gene causes a defect in the heart, and it does. So what we have to do is look for those defects. That we would like to figure out what's going wrong, so that maybe in the future some not cure, but we'll understand how our protein functions better, and then maybe that knowledge can be used, say, for curing a defect or curing cancer, because our gene is also involved in some types of cancer, B-cell lymphomas in particular. So if we can understand better how our protein works at a basic level, like in a normal situation, then you can compare it to what's abnormal, and then try to figure out if there's a way that you can alleviate that or fix it or cure it. That's the ultimate goal, but what we do is called basic research. So it's trying to understand under a normal condition what does our protein do? Like what its function is. I come in and there's usually a few things to do. So like I said, I take care of mice and work with mice. So basically I have to go to where the mice are taken care of, and I have to check on them because sometimes I have to do a dissection. So I'll have to go up and get a mouse and bring it down here, and then I'll do a dissection down here. Another thing that we do is, so I take care of all these mice, and I have to breed them together and then verify what their genetic makeup is, to see if they're a mutant or if they're a wild type, because you can't always tell what their genetic makeup is. So what I have to do is take some of their DNA. But to do that, what I have to do is I take a little bit of their tail, the very tip of their tail, and just clip a little bit off. And what we do is like we eat away I use an enzyme that breaks down the proteins. You precipitate out the DNA, and then you spin it down so you get like a pellet, and then you resolubilize that, and then I do what's called a polymerase chain reaction. So it's a way of specifically identifying which combination of genes that particular mouse has. Sometimes I have to make those solutions. So in that case, I would go over to the chemical bench and make that solution up. We have to sterilize things. So we have what's called an autoclave. I don't know if you've learned about an autoclave, but so solutions like this, sometimes they need to be sterile, so that there's nothing in there that's going to grow or degrade what I'm doing, like my DNA and stuff like that. So we can look at the cells then and see what our protein is doing. And then actually one of the ways that we can do that is in bacteria, what we can do to our gene is add things to it like tags. So we can add fluorescent tags onto it, so that when it does get put into a cell, it'll generate like a green color, a green fluorescent color. So then you can track where your protein is and see if it goes to the nucleus or the cell membrane. Yeah.
That's great.
So the way that we prepare these skeletons is I dissected, for the most part, most of the tissue off. And then what we did was we went to a lab in St. Paul, on the St. Paul campus, that have these beetles called dermestid beetles, and they actually are flesh-eating beetles. And so you put the skeleton, or the partially dissected, you try to get most of the flesh off, but then you put them in with these dermestid beetles for a week or two or something like that, and they just chew away the flesh, but they leave the skeleton.
Wow.
That's pretty cool. It's kind of like using biology to help your biology experiment go on. So it's kind of cool. So yeah, what we do then is try to gather data about the differences of the phenotypes that occur, and then we gather that data and try and put it together to do a publication in a journal, in a scientific journal. And so that's kind of the goal for us, is to get publications, and then as you get publications, then you apply for another grant, or my boss applies for grants, and then if we get funded, then I get paid under that grant. So it's the way that we sort of make things work is through NIH grants, which is a federal funded grants. So it's independent from like private companies and things like that. The best advice I would give is to get any experience you can, and that would be most likely sort of a volunteer basis. I would say when you're younger, when you're in high school and early college, I think that at least donating your time to some extent just to get your foot in the door somewhere is really worth your effort. It's just to come in and learn the different glassware, like what is the different glassware, whether you're or if you can find a job where you wash glassware, dishware for a lab, and just getting in a lab environment to understand like what the environment is like, to see whether or not that's for you, because not everybody likes labs. Another option is, if you're quite good at science, if you're really, really interested, you could actually contact, usually by email, a professor, like at a university, and say, for a summer, say, I'm really interested in science, and I'd like to get some experience in a lab, and I've done this, this, and this. I've done PCR. You guys said you did PCR in class. I mean, definitely list things that you've learned in class, contact, maybe say, for example, if you're interested in genetics, contact a professor, or a few professors that you're interested in at a university, college, whatever. Just say, I would like to get more experience, and can I spend some time in your lab in the summertime? And some will say no, but some might say yes. And all you need is that one person to give you sort of a little bit of experience to build on. Because once you go to college, I think it's also important, if you're going to be in science, to get a job, whether, again, if it's dishwashing or making solutions, pouring plates, kind of things that kind of the upper level people in the lab need help with, but it's a good way to start learning in the lab. Because once you're there, a lot of times people are willing to sit down with you and explain what their project is. Oh, do you want to help me do this? Do you want to learn how to do restriction digest? Do you want to, so it's a good way to just get in there somehow and get a little bit of experience. Oh, the other thing would be, if you are going to go to like a particular professor and contact them, I would say the best thing to do is look up what their research is. And even though you don't understand it all, I would say try to read a paper of theirs or a couple papers of theirs to understand at least kind of what they're doing, because that makes a good impression with a professor. They will be a lot more impressed if you've read and you know a few words from what they're doing in their research. Will make a good impression on them. So that will oftentimes give you that little edge to get in the lab.